Welcome to the lecture today for the topic of structural lipids. We talked earlier about storage lipids. This will be a little bit different in that we're going to talk about how lipids are used in membranes primarily. Before we were talking about how lipids were used as energy, and today we'll talk about how they're used in the storage in membrane, or how they're used in membranes. So a couple things that you're going to want to know about lipids, or you're going to want to focus on lipids. You're definitely going to want to know and understand how lipids are named. So we talked about that in the previous lecture. You're also going to want to know about the polar head groups, and in particular the structure. So today when we talk about structural lipids, definitely want to talk about that. We'll want to talk about how the fluidity of a membrane is affected by the type of fatty acid by its fatty acid composition. We'll talk a little bit more about that when we talk about membranes, but we're going to talk about that. We want to definitely focus on that as we get going in through these. I'm just going to give you an idea of some of the things that are going to be important for the exam and things that you're going to want to definitely study as you go forward. So those are the main ones. What else do I have that we need to focus on? Talk about which fatty acids have the highest melting temperature. So if they have a kink, that's going to decrease the melting temperature. In particular, cis fatty acids are going to cause that melting point to increase. You're definitely going to want to know, I mentioned it just earlier, you're definitely going to want to know about the head groups. And you're definitely going to want to know about the backbone molecules, so whether it's a glycerol, a sphingosine, and we'll talk about all these today. But those are important things you're going to definitely want to know as we get going on this too. And then when we talk about lipoproteins and cholesterols, we'll definitely want to focus on that. Okay, so those are things that are going to be important today to focus on and remember, especially for the exam. Okay, so some announcements. Major class goals. So for us, this is the third time I've taught biochemistry in a general type of way, and we have a couple of goals. First off, I just want you to know biochemistry. I think it's a really important. That's why we're incorporating the ACS exam, so you know that you have the information. Next off, it's a good tool to have in your tool set. A toolbox, so to speak. As you get going into pharmacy, you're going to want to know things, and this provides you the opportunity to go and investigate those things. Hey, what's the mechanism of action of acetaminophen? What's the mechanism of action of a statin? What's the mechanism of action of Repatha, or any of Humira? All these kind of things, and this provides you the background information. Along those lines, the other thing that's really important for us is, and we hear this all the time, and I'm not even in the department, so I know it's a big deal, is that as pharmacists, as pre-pharm students, you are not prepared in biochemistry to handle some of the topics that are going to be discussed from you. And there are certain professors, and we're not going to name any names, but there are certain professors that teach in the pharmaceutics program who are constantly on us and on about chemistry, about how unprepared the students are to take those classes. So our goal is to get you to be prepared, and I don't know, we'll see how this works for the first time, and we'll obviously this is kind of an experiment because this class is not a traditional class. But those are the major goals of what we're trying to accomplish, and along those lines, as a tool in the toolbox, one of the things that I wanted to do was do the PIMOL thing a little more to see how that worked, and it needs to be revamped in my book for this format. This format's a little different than what the 460 or 260 classes were, and so what we're going to do from here on out is I'm going to cancel the other two assignments, and we're going to take the two that we've done and we'll double their value. And I want you guys to focus the last 4 weeks on getting the material down. In the future, we'll revamp that program and see if we can make it a little more effective. Maybe it was just me being gone more than I want. When they asked to teach this class, I already had my summer laid out, and I continued to offer to help because of the nature of the course and some of the things we wanted to accomplish. my travel schedule this summer is crazy, and it's not conducive to this class, and maybe that has an impact on the video. And I don't want to punish you for that, my lack of being around. So we're going to open up the last half of your class. You've done a great job with the PyML at this point. I'm relatively happy with what we have. We'll open up the rest of the class, and we want you to focus on getting the material down. This week's a little more just straightforward memorization. Last week was math intensive. Coming week's just going to be math intensive. So be prepared as you go forward. There's some weeks that are just memorization intensive. This isn't as memorization intensive. It's pretty straightforward material, but there will be points at which it is a little more intensive. Okay, we're gonna have office hours this week. I am out of town doing a review session with the NIH, and so a study section, meaning I review grants for them in DC, and so on Monday and Tuesday. So I will be available starting Tuesday afternoon to help you. I'll try to put some time aside on Monday night, although that may be difficult. I will put that on, and I'll be on Blackboard in the discussion group. So that'll be the mechanism by which you can interface with me. I apologize, I didn't realize my dog was, our dog was barking so loudly. You probably still hear him. All right, so what are we talking about today? We're talking about lipids, and we're going to talk about the structural lipids today. That's an important component, a major component of the functions of lipids, and today we're going to dive deep into that. So as a background, and this is, DDT, you're probably all familiar with DDT. DDT was a colorless, crystalline, tasteless, almost odorless organochloride, and it's known as an insecticide. And it's also known for its environmental impacts. So after World War II, DDT was used as an agricultural insecticide. And it was fairly widely distributed. It was fairly widely used. And here's the chemical structure of it. You can notice it has very unique chemical structure. In fact, it was deployed, and the inventor of the pesticide, Paul Mueller, who was a German, won the Nobel Prize for it because of its impact. But in 1962, Rachel Carson came out with a book called Silent Spring, and from there was documentaries made, there was testimony in front of Congress, there was a foundation set up, and from there things started going downhill for DDT. And then by 1972, the ball started rolling, and DDT was banned by the EPA. And there's one property in particular of DDT that was pretty important. The way DDT works is it's an acetylcholine receptor blocker, and so it messes with your neurons. And you can see up in the left-hand corner, this is pyrethroids and DDT block the sodium channels that are associated with acetylcholine receptors. We'll talk about acetylcholine receptors a little later in the course. And that's a good thing. It doesn't affect all acetylcholine receptors, and there was an appearance that it didn't affect any human receptors, but that came out to be untrue. What was the problem? Okay, so and all things are not clear, right? So obviously mosquitoes are a massive carrier of malaria, dengue, yellow fever, Zika virus. So in 1930s there was without DDT there was widespread mosquito growth. By the 1970s the DDT had killed off a large chunk of them. Back by 2011, mosquitoes are everywhere again. So that sometimes it's not as clear cut as we make it out to be. Believe me, I'm not advocating for DDT use. I'm just saying that there are consequences that we have on both sides. So malaria and some of these other viruses have increased because we've cut back on DDT. So what's the big problem with DDT? And if you were in class, I'd give you a couple minutes to research this. But since we're doing this video, DDT is lipophilic, meaning it's soluble in and accumulates in fatty acid. And then so things like fish and human fatty acids or fatty tissues will absorb DDT and it'll keep it in. Other pesticides that that we use, such as glyphosate, which is Monsanto's, which is very, very common. you're probably familiar with is Roundup. They're metabolized and excreted, and the concentration is not felt because it's excreted and it's just redistributed. The other problem with this is biomagnification, meaning that it concentrates in our food supplies. So let's take this, and as we get to the end of the lecture, we'll talk a little bit more about DDT. But let's get into the lipids and why lipids are important. So earlier we described lipids. At the bottom part of the slide, we characterized lipids as being characterized by two distinct types of chemical functional groups, ketoacyl and isoprene groups. Another way we can classify it, another way you'll probably find it on exams, hint hint, is that it's insoluble in water. But it is soluble in organic solvents. So both of those are used as classification mechanisms for lipids. Lipids are very heterogeneous, meaning, and they're naturally occurring, meaning that they commonly have all sorts of different properties. Structural lipids, which are a little different than storage lipids, contain polar head groups and non-polar tails. Obviously, very commonly, structural lipids have attached fatty acids. And the diversification of structural lipids comes in multiple forms. First, meaning there's a modification or a different backbone. You can change the fatty acids. There's sometimes fatty acids are found in certain positions, and we'll discuss those a little bit. And we can modify the head group. We can also modify the attachment properties of those head groups to the fatty acids. The properties of the head groups generally determine the surface properties of the membranes, and so that's commonly why you see certain structural lipids in certain places. Different organisms have different membrane lipid head group compositions based on the function. That's related obviously to the structure and then the function, and different tissues have different membrane lipid head group compositions as well. So where we need different structure and functions of those, we will have different compositions of structural lipids. Also, inner and outer membranes can be different, and even within a cell, different types of membranes have different properties. Alright, so this is a revisiting. We've already talked about all the storage lipids, the triacylglycerols. Now we're going to go through today and talk about phospholipids, glycolipids, and ether lipids, each one in some detail. Now, the important difference between these two, as you might notice in this chart, I would actually memorize this chart, and I'll reemphasize this later, but the difference is there's a couple differences. Primarily, the first and most important is the backbone. We have glycerol, sphingosine, and you'll see ceramide in there as well. We also have the attachment, whether it's a phosphate attachment, an ester attachment, what have you, of certain molecules. And then we also have our polar head groups can be different. Also have an ether linkage is in there primarily. So complex lipids. So there's two main kinds. We'll talk about the ether lipids as well, but the complex lipids have two main kinds. Phospholipids, which contain an alcohol, two fatty acids, and a phosphate ester. In glycerol phospholipids, the alcohol is a glycerol. In sphingolipids, the alcohol is a sphingosine. In glycolipids, we have complex lipids that contain a carbohydrate. That's what the glyco means, and you'll definitely want to know that and how that impacts things for the exam. Two other classifications that you might see that are sometimes prevalent are lecithins and cephalins. These are two types where the first type, a lecithin, has a choline head. So we have a glycerol type backbone, and we have a polar head choline group. We have the same thing with the cephalin, and we have an ethylamine head group. So lecithins and cephalins are highly abundant in brain and nerve tissue. They're also found in egg yolks, wheat germ, and yeast. These are usually phospholipids, and they're composed of phosphoric acid with a choline, glycerol, or other fatty acids. And glycerol phospholipids in lecithins are there's multiple types, and they include phosphatidylcholine, phosphatidylethylamine, phosphatidylnositol, phosphatidylserine, and phosphatidic acid. And we will see these again. These are two different terms for classification, classifying common fatty acids. So let's talk about phospholipids. Phospholipids are a very broad class of lipids that are a major component of cell membranes. They form lipid bilayers because of their amphiphilic characteristics, meaning you have a polar head group and you have fatty acid that's fatty acid chains that are very, very nonpolar. Again, phospholipids are composed of two main groups: glycerol phospholipids and sphingolipids. We have a choline, which is a polar head group. We have a glycerol backbone, and then we have fatty acid chains that are in there. So glycerol phospholipids. Let's talk about those. Those are a major category of phospholipids. Basically, we take glycerol 3-phosphate. It's the backbone of phospholipids. Glycerol is itself is not chiral, but it has a definite plane of symmetry through the C2 carbon. So if you add any other functional group typically to it, it becomes chiral. So that's prochiral. This structure is very common. So we have our glycerol head group followed by a phosphate group, and that provides the backbone of the lipid. So the common form of glycerol 3-phosphate and phospholipids is this glycerol 3-phosphate in the R. It has the R configuration, which is what you see. Alright, glycerol phospholipids, again, just kind of gives you a general structure. So we have glycerol. We can put any number of fatty acids on here. In this situation, they have palmitidic acid and oleic acid. An oleic acid, which is a unsaturated fatty acid, is commonly found in the C2 position of this glycerol. And then we have our phosphate head group. And that phosphate head group can also be a phosphate linkage, which you'll note here that we can also put amino alcohols there. We notice we have two negative charges on this glycerol phospholipid. The general structure of a glycerol phospholipid, again, we have our fatty acid chains, our glycerol, our phosphate linkage, and then we have a substituent head group. This can be any number of things. It can the unsaturated fatty acid is commonly found in that C2 position. This polar head group, we can put things, any. So here are the any number of things there. Here are the 5 major glycerol phospholipids. Phosphatidylserine, if we put a serine on for that amino alcohol. Ethylolamine is phosphatidylethanolamine, again acetylene. Phosphatidyl choline, lecithin, phosphatidylinositol. We'll talk about that. That's the basis for a GPI anchor. Phosphatidylglycerol. Those are all glycerol phospholipids. Here's some examples of a chart and their net charges. Phosphatidic acid, which just has a hydrogen for the X group, has a -2 charge, all the way up through phosphatidylinositol has a -4 charge because of that strong negative charge that is on the carbohydrate group in there. So this just gives you an idea of what the charges are, some of the functional groups that were added in as a X. As a polar head group of the glycerol phospholipids. So let's go into detail on some of these other ones. Phosphatidylserine is a phospholipid, and it's a component of a cell membrane. It plays a key role in signaling. In particular, it's important in apoptosis. So this is commonly found on the inside of extracellular, and the cytosolic side of a membrane, and flipase will flip when a cell is healthy, it'll flip it back over to the inner membrane. When it gets to be unhealthy, it'll start flipping this on the outside. You will note that it is also used as a role in blood coagulation. When circulating platelets encounter injury, this is usually externalized. Phosphatidylserine is externalized, and it provides the mechanism of action by which blood clotting and other repair mechanisms are initiated. It's usually abbreviated PTD-L-SER or just simply PS. That's phosphatidylserine. You can see we've got our 2 fatty acid chains, we've got a glycerol backbone, we have a phosphate linkage, and then we have our amino acid serine, where the functional R group is an alcohol, and that provides the linkage, and we have our amino acid. We have an amino head and a carboxyl head. Phosphatidylcholine, very similar structural formula. We have our 2 fatty acids. Our backbone is glycerol. We have a phosphate linkage, and then we have choline in as the polar head group. Phosphatidylcholines incorporate choline as their head group. They are, again, a major component of biological membranes. They are easily obtained from diet. They're also a member of the lecithin group, which we talked about earlier. And phosphatidylcholine is also very important as a lung surfactant. Otherwise, so it if something deflates, 10 times the normal pressure is needed for reinflation, and as a surfactant, phosphatidylcholine allows less pressure to be initiated when you're reinitiating reinflation. So phosphatidylcholine provides an important biological role for that. Phosphatidylethylamine, class of phospholipids found in biological membranes. They're synthesized pretty readily from diglycerides and releasing of CMP. Phosphatidylethylamine is found in the inner leaflet of a lipid bilayer. It's found in all living cells. It's about 25% of all membrane fatty acids, but you also find them also a lot in the nervous system, where they can make up to 45% of all phospholipids. It's abbreviated PE. It's also implicated in membrane fusion as an important element. So let's switch over to sphingolipids and discuss those. Sphingolipids have a sphingosine backbone. Sometimes it will be modified, and on the left, you'll see that on the sphingosine on the left. Sometimes it's modified to be a ceramide, which you'll see in the middle. Something like sphingomyelin is an important sheath lipid that you will find in the myelin sheath. These are long-chained amino alcohol. Or sphingosine is a long-chained amino alcohol, and that's what everything is attached to. I think it's important to note there is no glycerol in this molecule. And you do still see linkages, but these linkages are amine linkages, as you see with the example here. These are sugar-containing glycosphingolipids, or I should say sometimes you see these. If you have a glycosphingolipid, those are largely in the outer membrane of the plasma membrane, and we'll talk about those in a second. So here we have sphingolipids. Again, we have a sphingosine molecule, we have a long-chain fatty acid, and then we have a polar head group with a oxygen linkage. Ceramide has H as the head group. Sphingomyelin, which is found in myelin sheaths, or phosphatidylcholine is found in those locations. And then we also have some of our gangliosides, which are complex oligosaccharides that are found linked on the head group substituent. Again, glycosphingolipids, those are found in the exterior cell where we attach sugar to. Sugars have multiple functions within these, including charge and interactions with as a signaling mechanism, as a receptor mechanism. Cerebrosides have a single monosaccharide, but multiple cerebrosides obviously are small chain, but there are also numerous longer chains that can also be added. That small chain is usually glucose or galactose. And you can see from here we have a sphingosine backbone with a fatty acid chain, and then we have our sugar or carbohydrate residue with a linkage. A ganglioside, again, can have one or multiple oligosaccharides. You'll definitely want to know this for the test. They're abundant in cell membranes of neurons. They're, again, on the outer surface. They usually act as a receptor for hormones. And we'll talk about this in a little bit, but accumulation in this situation of the ganglioside GM2 can cause Tay-Sachs disease. Again, important note, we can have one or multiple sugar residues that are important in cell organization. Gangliosides, just as a quick note, can have multiple. In this situation, you have glucose, galactose, and other sugars in a residue, as well as you have your sphingosine backbone and then you have your fatty acid chain. It's implicated in multiple disease states such as HIV. Glycosphingolipids are also important in blood groups, and we're just going to mention them here. I'll talk about them more later, but notice that we have a ceramide / sphingosine functional backbone with fatty acid, and then we have a carbohydrate structure. That carbohydrate structure provides the differences for the different blood groups. Again, something we'll talk about more as we get going into this week, because it's also related to carbohydrates. Sphingomyelin, again, we have a sphingosine molecule with a fatty acid chain, and then we have phosphocholine as the polar head group. You'll see those in, again, a lot of different places, including the neural cells. Sphingomyelin is essentially a myelin sheath protector. So ceramide has, again, a fatty acid and then a sphingosine linkage, or potential sphingosine as a fatty acid. Glycolipids are very common. They're a complex lipid that has carbohydrates and ceramides. The carbohydrate is very commonly glucose and galactose, and you can have mono or oligosaccharides in this situation. Cardiolipin. Cardiolipin is an intermitochondrial membrane. It's important in maintaining the respiratory complex. Again, you can have multiple fatty acid chains. You have, you have your ceramide, and you have two phosphate linkages that are complex and put them that have fatty acids attached to them. Excuse me. Last but not least, in the major classifications of fatty acids, we have our ether lipids. Plasmalogen is one. We have plasmalogen that's linked to a fatty acid by ether linkage, and on that we have a polar head group that has ethylamine with a phosphate linkage. So plasmalogens obviously have very important charged head groups. Along those same lines, platelet activating factor has this very similar structure, and then we have an ether-linked alkane with platelet activating factor. We have acetyl ether as a group, and then we also have a phosphate linkage with a choline as a group that provides specific structure and function based on the charge and the size of that molecule. One important note that we haven't discussed is turnover of membrane lipids is very, very important in mitigating diseases, and if we have trouble turning them over, recycling them, then we get a whole bunch of diseases, including gangliosidosis, Tay-Sachs, Gaucher's, Sandhoff's, Fabry, and Niemann-Pick. So if we don't get these molecules turned over, recycled basically, then we are going to have problems. As a side note, waxes are ethers. They're also a lipid. They're ethers that are combined, waxes are ethers that are combined into a long chain fatty acid, usually between 14 and 30 carbons. Plants produce on their surface to minimize loss of water, and animals use them on their fur to maintain their water proofing capability. We also use them commercially for any number of sources, and they have important structural formulas that you can see here below. Phosphatidylinositol is an anchor protein, and it's a glycoprotein. It has a carbohydrate structure. That carbohydrate structure also has phosphate linkage opportunities, and it provides the ability to anchor a protein in a membrane. This is kind of the example of what you'd see, and we'll go into greater detail on this when we talk about carbohydrates, but that phosphatidylinositol is the lipid that goes inside the membrane. We have a phosphate linkage. We then have a huge carbohydrate linkage, again another phosphate linkage, and then that goes to the protein. So that allows for a protein to be anchored in the surface. And then commonly you'll also see that protein has carbohydrates on it as a post-translational modification, and so what goes around comes around. You'll note that the little blue squares and little green circles are in essence the same thing as this linkage, this carbohydrate linkage, just different notations. Okay, let's bring it all back together and tie it all together, and we'll talk a little bit about GDT. So, complex lipids, this is what we're talking about today. Simple lipids are used for storage. Complex lipids are used as membrane substituents. So, this slide is very important to know. Two other important concepts surrounding this. Number one, if you look at DDT will slide up in all of the location. It's lipid soluble, so it will go up in all the locations in a membrane and hide out, and that allows it to bioconcentrate, and that bioconcentration can be deadly for people who are not able to deal with it. I would know this chart for the exam, know the general structures of all these different fatty acids, and I would know general examples of each one of these lipids. Hope you enjoyed. I look forward to seeing you later in the next lecture.